Enzyme activity. Catalysts increase the rate of chemical reactions, but are not used up in the process. Although actually participates in a chemical reaction, it's not permanently modified and may be used again and again. And so this is one of the reasons why using catalysts is so popular now, because it's environmentally friendly, right? The catalyst gets recycled over and over. And now enzymes are true catalysts that lower the activation energies, thereby allowing chemical reactions to achieve equilibrium more rapidly. And so if you take a look at this reaction coordinate diagram that's shown here in figure 12.22, we take a look at our uncatalyzed reaction, you see that the activation energy is very high, right? There's a big barrier that you have to get over in order for the reactant to be converted into the product. However, when you put in the catalyst, what it does is it lowers the activation energy. So this lowers the activation energy, and you can see that the barrier that you need to get over now is much smaller. And so since the activation energy is smaller, it means the reaction is going to occur faster. Now, many of the important enzyme catalyzed reactions are similar to organic reactions that we studied way back in chapters 8, 9, and 10. If you think about things like ester hydrolysis or alcohol oxidation, however, lab conditions cannot match what happens when these reactions are carried out in the body, where enzymes cause them to occur under mild pH and temperature conditions. Also, enzyme catalysts within the body can accomplish in seconds the reactions that ordinarily take weeks or even months under laboratory conditions. And an example of that, of the influence of enzymes on the rate of reactions essential to life, is the process that removes or that moves carbon dioxide, which is a waste product of cellular respiration, out of the body. Before this can be accomplished, the carbon dioxide must be combined with water to form carbonic acid. And you see the reaction shown down here in the bottom right-hand corner. In the absence of the appropriate enzyme, the carbonic anhydrase, the formation of carbonic acid takes place much too slowly to support the required exchange of the CO2 between the blood and the lungs. But in the presence of the carbonic anhydrase enzyme, this vital reaction proceeds very rapidly. So each molecule of the enzyme can catalyze the formation of carbonic acid at the rate of 36 million molecules per minute. Now, enzyme activity, and you see, you need to know this definition, refers to the general catalytic ability of an enzyme to increase the rate of a reaction. Now, this amazing rate of 36 million molecules per minute, at which one molecule of carbonic anhydrase converts CO2 into carbonic acid, is called a turnover number. Okay, so the number of molecules of substrate acted on by the molecule of the enzyme per minute is the turnover number, and it's one of the highest known for enzyme systems, this 36 million molecules per minute for the carbonic anhydrase. More common turnover numbers for enzymes are more like 1,000 reactions per minute. Nevertheless, even such low numbers dramatize the speed with which a small number of enzyme molecules can transform a large number of substrate molecules. And if you take a look at table 12.5, and table 12.5 is shown here on this slide, it lists the turnover numbers of several different enzymes. So we see our carbonic anhydrase has this huge number here, okay, of 36 million. And then you look at something like chymotrypsin, which is lower. So is 6,000. And chymotrypsin hydrolyzes specific peptide bonds. And then if you look at something like lactate dehydrogenase, you see that's around 60,000. So again, the carbonic anhydrase is a very high turnover number. Experiments that measure enzyme activity are called enzyme assays. Assays for blood enzymes are routinely performed in clinical laboratories, and such assays are often done by determining how fast the characteristic color of a product forms or the color of a substrate disappears or decreases. Reactions in which protons are produced or used up can be followed by measuring pH and how pH changes with time. Because some clinical assays are done many times a day, the procedures for running the assays have been automated and computerized. The determined enzyme activity levels are usually reported in terms of enzyme international units. So you need to know this definition. And enzyme international units, or IU, which defines enzyme activity as the amount of enzyme that will convert a specified amount of the substrate to a product within a certain amount of time. Now, one standard enzyme international unit, so 1 IU, is the quantity of enzyme that catalyzes the conversion of 1 micromole of substrate per minute under specified reaction conditions. Thus, unlike the turnover number, which is a constant characteristic for one molecule of a particular enzyme, international units, the IUs, measure how much enzyme is present. For example, an enzyme preparation with an activity corresponding to 40 IU contains a concentration of 40 times greater than the standard. And this is a useful way to measure enzyme activity because the level of enzyme activity is compared with normal activity and is significant in the diagnosis of many diseases. Although enzymes differ widely in structure and specificity, a general theory has been proposed to account for their catalytic behavior. Because enzymes molecules are large compared to the molecules whose reactions they catalyze, meaning the substrates, it's been proposed that the substrate and enzyme molecules come in contact and interact over only a very small region of the enzyme surface. And the region where they interact is what we call the active site. So again, an enzyme is a very big molecule, then your substrate is going to be much smaller. So the substrate is only going to react at one very small part of the enzyme, and that's what we refer to as the active site. So the binding of a substrate molecule to an active site of an enzyme may occur through, it could be a hydrophobic interaction, it could be hydrogen bonding, it could be ionic bonding, like a salt bridge kind of thing. And now the complex formed when an enzyme and a substrate bind is called an enzyme substrate complex. And once the complex is formed, the conversion of the substrate to the product can take place. So you see the general reaction shown here where you have your enzyme, then your substrate. They come together to form the complex, so the enzyme substrate complex, and then the substrate gets converted into the product by the enzyme, and then the enzyme releases the product. And so now your enzyme, you see that you end up with your enzyme again. So the enzyme is, can go right back to the beginning. So it can get recycled over and over. And specific example that they used here is sucrase. So if we take sucrase, which is an enzyme, and we combine it with sucrose, which is table sugar, it forms the sucrase-sucrose complex. And what two sugars come together to form sucrose? Well, it's glucose and fructose. And so you see that glucose and fructose get released as products. Oops, let me get my highlighter. They get released as products, and then your sucrase gets reformed so it can go right back to the beginning, right? So it can catalyze another hydrolysis of sucrose into glucose and fructose. Now, the chemical transformation of the substrate occurs at the active site, usually aided by the enzyme functional groups that participate directly in making and breaking of chemical bonds. And after the chemical conversion has occurred, so once the product is formed, again, like I said, the product gets released from the active site, and then the enzyme is free for another round of catalysis. Now, there's a couple theories about how enzymes and substrates bind together. And according to what we call the lock and key theory, which is the oldest of the theories, the enzyme surfaces will accommodate only those substrates having a specific shape and size, and thus only specific substrates fit in a given enzyme and can form a complex with it, just like a lock and key. And so you see that down here at the bottom, okay, you see that you have this specific shape of your active site here in your enzyme, and then this would be your substrate here. So the substrate fits in here just right. So you see how they match each other just like a lock and key. Now, a limitation of the lock and key theory is the implication that the enzyme conformations are fixed or rigid. So that would mean that the enzyme can't morph or change its shape at all. And research suggests that the active sites of the enzymes are, in fact, not completely rigid like a lock, like a metal lock. And so a newer theory is the induced fit model or the induced fit theory, which is a modification of the lock and key. And it proposes that enzymes have a somewhat flexible conformation at the active site that can adapt to an incoming substrate. And so the active site has a shape that becomes complementary to that of the substrate as the substrate binds to the enzyme. And you see that right here. You can see that the open or the active site here that's opened up, you can see the shape isn't exactly the same as that of the substrate, but when the substrate goes into the active site, then you see it adopts the shape. So you see how the shape of the active site changed over time. And so that's what we call the induced fit theory, which is, I believe, more widely accepted now. It's a more modern theory. Now, if you remember back in section 12.3, that proteins can be simple, they could have just a couple of amino acid residues, or conjugated with some kind of prosthetic group, like a heme group, like we saw in hemoglobin. Many enzymes are simple proteins, whereas many others function only in the presence of specific non-protein molecules or metal ions. If these non-protein components are tightly bound to and form an integral part of the enzyme structure, then they're true prosthetic groups. Often, however, some non-protein component is only weakly bound to the enzyme and is easily separated from the protein structure. This type of non-protein component is referred to as a cofactor. So cofactor is a non-protein molecule or ion that's required by the enzyme for catalytic activity. So it's like, something that you need for the enzyme to be able to act. And when the cofactor is an organic substance, then we call it a coenzyme. So not all cofactors are going to be organic. Okay. Again, sometimes it could be a metal ion even. So a coenzyme is going to be when it's an organic molecule that's required by an enzyme for its catalytic activity. And the cofactor, like I said, may also be an inorganic ion, like a metal ion, like the iron 2 found in the heme molecule that's found inside the hemoglobin. So the protein portion of the enzymes requiring a cofactor is called an apoenzyme. So apoenzyme is a catalytically inactive protein formed by the removal of the cofactor. So you can see here this equation they've shown you, which is if you take the apoenzyme and the cofactor, you get an active enzyme when you put it together. A typical inorganic ion, so you see the word here, or you see inorganic ion shown here. Typical inorganic ions are going to be ions like magnesium, which has a 2 plus charge, or zinc, or iron with a 2 plus charge. Okay, those are very common, typical inorganic ions. And for example, the enzyme carbonic anhydrase that we saw earlier on only functions when zinc 2 plus is present, and renin needs calcium 2 plus in order to curdle milk. So numerous other metal ions are essential for proper enzyme function in humans, hence they are required for good health. Like metal ions, small organic molecules that act as coenzymes bind reversibly to an enzyme and are essential for its activity. An interesting feature of coenzymes is that many of them are formed in the body from vitamins, which explains why it is necessary to have certain vitamins in the diet for good health. And you can see a bunch of different vitamins here, and you see their coenzyme forms.